Am I? Is there any voice? Is everybody? Can anybody hear me?
Yeah, we can hear.
Suddenly saw everything went quiet, so I was kind of worried. Ki kya ho
Hi,
gaya?
actually, when I was worried.
Yeah, I know. I was like, shit.
Yeah, we are now on YouTube live, so I think everybody.
Yeah, we are live.
Okay. All right. All right.
Okay, good evening to everyone. Respected Principal of VAS College of Arts, Science and Commerce, Dr. Anita Kanwar. Invited speaker of the webinar, Dr. Subhojit Sen. Faculty members of microbiology from our college and colleges across Mumbai. All our esteemed alumni of the microbiology department and dear student participants. Inspire Me is an initiative of VAS College, Department of Microbiology, that seeks to spread knowledge by organizing talks and interactions with PhD scholars, scientists, and professors from premier research institutes. As a part of this series, the Microbiology Department of VAS College had organized the first webinar on the 23rd of May on arboviruses and was delivered by Dr. Finny Vergis, postdoctoral fellow, Rabord University Medical Center, Netherlands. This time, we have come up with another interesting discussion on defining epigenetics, heritable punctuations in the DNA language. I now request Vice Principal and Head of the Microbiology Department, Dr. Shantini Nair, to deliver the welcome address. Over to you, ma'am.
Thank you so much, Shweta. Respected Principal of VES College of Arts, Science and Commerce, Dr. Anita Kanwar. Invited speaker for the webinar today, Dr. Subhojit Sen. Faculty members of microbiology from our college as well as colleges from all over Mumbai. All our esteemed alumni members of the microbiology department and dear student participants from our college as well as from colleges far and wide. Good evening and a very warm welcome to all of you. A special welcome to our speaker for today, Dr. Subhojit Sen, who is a very prominent alumnus from our department. I'm truly happy to announce that all his teachers who have taught him along with me have joined us for this session today. On behalf of the department and our college, I extend a very warm welcome to all our former staff members, including our honorable former principal, Dr. Jayashree Fadnis, and our honorable former HODs, right from Dr. Maitri Paul, Dr. Hema Vardarajan, and Dr. Nagesh Malik. We are extremely grateful to them for being with us today. I'm sure all of these teaching stalwarts would remember teaching Subhojit during his graduation days, where he was known for giving excellent seminars way back in 1995 that made a permanent impression in our minds. We were immensely happy when Subhojit Sen had bagged the distinction of being the top ranker at the University of Mumbai final examination in that year. And that was the first instance where our college had this honor. I am sure all his teachers, including me, become nostalgic thinking about his student days, where he would be full of pranks while at the same time an exemplary student and yes, an excellent singer. So both, we are really happy to have you with us today. The Microbiology Department of VES College of Arts, Science and Commerce takes pride in organizing the second webinar in its series today. In this period of COVID-19 lockdown, when I took over as an HOD, I am very glad to have had the wholehearted cooperation of all our present young staff members who are finding solutions to keep the teaching learning process on. All of us felt that the conduct of this Inspire Me webinar series on topics relevant to microbiology and the current scenario would be a step towards connecting with a large number of students, as well as to be able to cross boundaries across the globe to connect and receive from the best of resource persons so that the learning process continues. As Shweta mentioned, last week we had our first webinar with Dr. Finny Verghese on arboviruses, which was very well received and appreciated with over 500 participants. We in the microbiology department are truly humbled by your positive feedback and huge response, and hope this series of webinars would meet your expectations. The microbiology department at VES College over a period of more than 4 decades has seen a vertical growth, offering courses from BSc to MSc by papers, MSc by research, and since the year 2015, our department is also recognized to offer PhD courses in microbiology from the University of Mumbai. In fact, we have 7 PhD students who are still continuing their work under Dr. Nagesh Malik, our former HOD, who I'm sure are doing excellent research that will enhance the research culture of our department. Dear student participants and faculty members, we are still taking baby steps in understanding the technicalities of hosting webinars, and I plead to all the participants to bear with us for technical issues, if at all. But try we will to overcome these challenges. With the strong support that we receive constantly from our VES management and the dynamic leadership provided by our honorable principal, Dr. Anita Kanwar, and of course, the enthusiasm of all my colleagues. I sincerely hope that all of you would benefit from this learning exercise, and I'm sure this webinar too will be a most enjoyable one, knowing Subhojit's penchant for teaching. The COVID pandemic has suddenly compelled the world to start adopting a new way of life, wherein there will be a paradigm shift in not just the field of education, but in every sphere of life. The faster we all realize this and evolve strategies to cope with the new normal, the better prepared we would be to face life ahead. Not by just staying safe, but also keeping our spirits high, keeping ourselves mentally and physically fit. Dear students, remember that when you face difficult times, know that challenges are not sent from above to destroy you. They are sent to promote, increase, and strengthen you. Be tough, stay safe, stay healthy, stay blessed. Thank you, one and all.
Thank you, Shantini ma'am, for your encouraging message. Yes, ma'am, we really need to realize that our new normal and we should work towards this normal. Now, I call upon our principal, Dr. Anita Kanwar, to give her message. Principal ma'am.
Good afternoon, all dear students and the participants. Our esteemed guest and speaker for this webinar, Dr. Subhojit Sen from Center of Basic Sciences, University of Mumbai. Our former principal, Dr. Jayashree Fadnis, former HODs of microbiology departments. Our vice principal and head of microbiology department, Dr. Shantini Nair, other members of microbiology department, all invitees and my dear students. Very warm welcome to you all from Vivekanand Education Society's College of Arts, Science and Commerce, Chembur, Mumbai. Vivekanand Education Society was founded in 1962 by late Shri Hashuji Advani, a great visionary and educationist. Society presently runs 26 institutions with more than 22,000 students, with many higher education institutions like Institution of Technology, Architecture College, Law College, uh, uh, Pharmacy College, and Art Science Commerce College. Our inspiration is youth icon leader Swami Vivekanand ji. To give you brief introduction of our college, well, it is a multi-faculty college which was established in 1979. So we are around 40 years old. It is permanently affiliated to University of Mumbai, and currently our college is running 29 courses under aided and self-financing section. This includes 17 undergraduate programs, 9 postgraduate programs, and 3 PhD programs. Our microbiology department is also recognized for its PhD degree. With the help of our very supportive and progressive management, which is committed to the cause of education, and our dedicated teaching and non-teaching staff, hardworking students, we have achieved many laurels. To list few noteworthy ones, in our third accreditation cycle, we are again accredited by National Accreditation and Assessment Council with A grade in August 2017. We have received several awards from University of Mumbai, which includes Best College Award and the awards which our students have won. We are proud recipient of DST Feast Grant and DBT Star College Grant from Government of India. Microbiology department in our college has contributed greatly to the college by giving college the university top rankers, and today's speaker, Dr. Subhojit Sen, is one of the university top rankers in his batch. Few of our alumni members have acquired teaching positions in reputed foreign universities and center of excellence. They have always organized excellent seminars, and in today's context, I will say webinars for our students in the past, and today's program is not an exception. Dr. Subhojit Sen, our eminent alumni, talk on this occasion is very apt initiative and will surely provide an enriching insight for the participants. We all welcome you, Subhojit. I would like to end this speech by saying that all of you stay safe, stay healthy. Thank you.
Thank you, Anita ma'am, for your constant support and encouragement. We now begin with our webinar. But before that, a note to the participants. Once the session begins, you can type your questions in the comments section. The questions will be asked by the moderators after the talk. Also, a link for feedback will be provided in the comments section at the end of the session. Please ensure that you fill in your feedback since your e-certificate is linked to the feedback form. The link will be open for only 20 minutes after the session. So let us begin. We are not the victims of our genes, but we are in fact the masters. Think of our lifespan as a very long movie. Our cells are performers and are the essential members that make up the movie. The cell's DNA would be the script which would give instructions for all the actors, that is, the cells of the movie, to perform their roles. The DNA sequence encoded would be the words on the script, and the groups of these words that instruct key actions or events would be the genes. Did you follow the analogy so far? Good. The concept of epigenetics in this movie would be like directing. The script can be the same. But the director can choose to eliminate certain scenes or dialogues, altering the movie for a better or worse. After all, Ramesh Sippy's finished product Sholay would be drastically different if Rishikesh Mukherjee would direct it for the same movie script, wouldn't it? Similarly, in our body, why skin cells are skin cells and not neurons, even if they are essentially made up of the same genetic sequence? It means that in addition to traditional gene bases of inheritance, there exists something more. To make us understand this, in addition to genetic basis, we have with us Dr. Subhojit Sen, Ramalinga Swamy Fellow at CEBS, University of Mumbai, Department of Atomic Energy, Center for Excellence in Basic Sciences, Mumbai. I now request Dr. Donna Joseph to introduce Dr. Subhojit Sen to us and moderate the session. Over to you, Donna ma'am.
It is my privilege to introduce our speaker for the day, Dr. Subhojit Sen, who is one of our most illustrious alumni. Subhojit is both a scientist and educator who is interested in the epigenetic query of environmental triggers that push normal cells to become cancerous. He has had an exceptionally distinguished academic career, being a triple gold medalist. He did his BSc microbiology at VES College, topping Mumbai University. He went on to do his MSc in microbiology at the prestigious MS University, Baroda, where he once again topped the university. He then went on to do his PhD in molecular biology at TIFR, where he was awarded the Zeta Lobo Memorial Best Thesis Award in 2002 for his studies on DNA repair and recombination. Subhojit initiated his work in the field of epigenetics at the National Cancer Institute, NIH USA, followed by the Johns Hopkins Medical Institute, Tumor Biology, where he worked on translating some of his fundamental work into the molecular discovery of epigenetic pathways that drive gene silencing in cancer. He returned to India in 2013 as a DBT Ramalingaswamy Fellow at the University of Mumbai DAE Center for Excellence in Basic Sciences, where he heads the Laboratory of Epigenetics, which focuses on understanding stress-induced triggers in cells that eventually drive epigenetic disorders. He was awarded the 2019 Anil Sadgopal Cube Award for popularization of science and cultivation of scientific temperament. His work has been published in leading journals such as Cancer, Cell, Science, Molecular Cell, Natural, Structural and Molecular Biology, BMC Genomics, etc. Till date, his work has had more than 2,250 citations in peer-reviewed journals. Besides all this, Subhojit is now part of ISRC, that is the Indian Scientists' Response to COVID-19, which synthesizes literature from various scientific sources about COVID-19 and doles out the information in simpler words for the common man to implement in his daily life. Subhojit is a part of the core team that spearheads these efforts at hoax busting and fact finding Q&As about COVID-19. ISRC has been formed by Indian scientists and research students from different fields who have together built COVID-19 awareness resources in up to 18 different Indian languages. Dear Subhojit, we as college is proud of you and your achievement. We welcome you to this webinar and are eager to listen to you. Over to you, Subhojit.
Hi, thanks for that lovely, very flowery introductions. And I'm very happy to be part of this webinar series, and really happy that I'm able to do this in the times of COVID as well. So I'll probably start. I'll try getting into it quickly, and I'm going to try and keep this webinar kind of basic so that everybody who has some basic knowledge about epigenetics can understand most of it. Towards the end of the webinar, I will probably go a little bit into details, and I'm hoping that most of you should be able to grasp that those nuances. So with that, let me share the screen, and so I'm hoping that everybody can see the screen. Yeah. So can you see my pointer? Can somebody just can you see my pointer as well? My arrow?
We can see.
Okay.
Yes, sir.
So let me get into the seminar right away, and so the way I have structured the seminar is just to let you know that anybody who has some basic understanding and want to know about the fundamentals of epigenetics, essentially how does it work, and what does epigenetics mean, what is the basic molecular mechanism of this new field, is that's how I've structured the seminar. So that's what if you want to understand that, you should attend the seminar. And what kind of prior knowledge you need to know just basic molecular biology, essentially two main functions, which is how with the fundamentals of evolution and the central dogma. So if you understand these two fundamentals, you should be able to understand epigenetics from there onwards as well. So you don't need to know very high funda cell biology or neurobiology to understand epigenetics. You can appreciate epigenetics because it's one of the more fundamental aspects of biology. And what will you gain at the end of the seminar is that you will gain an appreciation of heritable processes outside of DNA, essentially because we know the genetic material is the heritable material. So then how is epigenetics allowing inheritance? And then the second thing is that how the environment can change us as genetic beings during our lifetime, right? So with that, let's get into the idea of where this comes from. So as we grow, as we start with a single cell embryo, from a single cell zygote, we become an embryo, and we become, we grow in our lifetime, and through this lifetime, we are exposed to different types of environments, stresses, as well as and when I say stresses, I'm talking about both biotic as well as abiotic stresses. And that is what essentially molds us as human beings. Okay. And what we know is that, for example, we know that from available data, what we know is that fluctuations in diet, early fluctuations in diet while you were in the mother's womb will affect how you have propensity to certain diseases later in your lifetime. Right, early fluctuations to diet can also lead to neurological disorders. But these are from correlations that people have drawn from what people experience during their lifetime, or studies that have studied what kind of stresses people go through their lifetime early on, and then what kind of diseases they become prone to. So, the question is that, can these environmental insults can they be tracked in a way, in a molecular way, so that then we can start understanding what predisposition we are getting to disease and how can we undo those predispositions, right? And another famous example is that of smoking, because we know that many people who start smoking at a very early age, but end up having cancer sometime in their mid 40s to early 50s. So their predisposition to cancer increases because of their smoking habits. Now, what recent experiments on mice and other model systems, mammalian models, have shown that mice can actually transmit this information, and mice, for very good reason, which I'll come to, are also mammalian models, and they can transmit this information across generations as well, right? And one of those key experiments was done where they fear conditioned the mice. They gave a particular signal to the mice that would make the mice fearful, and later on they showed that this fear conditioning property of the mother mice is passed on to the daughter and to the granddaughter as well. So essentially, and even though these even the daughter and the granddaughter never experienced that fear conditioning experiment, they still were fearful of those effects that the grandmother was fearful of. So this tells us many more things about how information can be transferred, because within a couple of generations, you don't expect such a large change of genetic material. So where is this change coming from? So the idea stems is that there is an insult during a lifetime, and the injury is completely separated from that insult. It's not that I get a bacterial infection and I suffer from disease. I start smoking and I get cancer tomorrow. And this is one of the major reasons why most smokers don't see cancer as a threat, because they don't see it happening in their immediate vicinity when they start smoking. So this separation of insult and injury essentially is what we're trying to understand. So so the hypothesis, therefore, is that there must be a transmissible cellular memory that passes on from early lifetime over onto your late lifetime, that remembers this information that you have already gone through, and that information is somehow predisposing you to disease, right? So with that understanding, let's now, and as I mentioned, that this predisposition also happens transgenerationally as well as intergenerationally. Okay, so without getting into further details, let me start as to how do we analyze a biological problem. Now, this is a very general approach that I'm taking. How to analyze a biological problem. I think most biological problems can be looked at with two essential lenses. Okay, one lens is the evolutionary lens, and the other lens is the central dogma lens. So the evolutionary lens tells you about how the disease might have evolved, tells us many more primitive things about the disease, whereas the central dogma tells us about the molecular nature of the disease, right? Tells us where the disease is acting, how it is acting, and so on and so forth. So this combinatorial look at any kind of biological problem that you choose will allow you to dissect that problem from many different angles. And let's take these two steps today. So to give you the first picture, this image that you're seeing on your screens is essentially what I call the Big Bang of life, right? Which started so Earth started at this time, which is about 4 billion years ago. And then when life started about 3.5 to 3.6 billion years ago, life has now evolved, and we are all today at this edge, which is the surface of this bubble, right? So we have started from this first common ancestor and then diversified into all these species that are now today existing on the surface of this bubble. What is important to appreciate is that any species today that we observe on the surface of this bubble has gone through the same amount of evolutionary time as compared to the bacteria or compared to the plant or fungi or mammals, right? So if you see that these are your grouped unicellular bacteria, prokaryotic bacteria and your archaebacteria, and then you have your plants, your unicellular plants to your multicellular plants, then you have the fungi, and then the rest are all your other amphibia and mammals and so on and so forth, reptilia, all the other kingdoms, right? So question is that what are these circles now? So these circles denote what we call as model organisms in biology. And why I particularly highlighted those circles is because these are the model organisms that I have worked during my research career. And today in my research career, I have finally stalled upon these two model organisms, which we study in the lab, which is Chlamydomonas and the human model system. So we shuttle between these two model systems and try and answer epigenetic questions. So how is it that I am studying a unicellular algae and trying to correlate mechanisms of that algae to what is happening in humans? How am I learning something about what is happening in humans, right? And that is precisely the concept of model organisms. The key understanding is that because we are all related, for example, if I'm studying a problem such as DNA replication, which happens in all organisms, because all organisms are DNA based, except for a few viruses, but they also go through similar cycles as far as the central dogma is concerned. So if I'm studying DNA replication, because it evolved really long ago, the same mechanism is used across all these model organisms, right? So therefore, whatever I learn from learning about DNA replication in E. coli, I can even apply that same concept to humans and learn something about human mechanisms as well. And this is how I shuttle back and forth between conservation of mechanisms. So understanding common mechanisms leads to helps us reduce the problem. We don't have to study complex problems from humans. We can reduce the problem to unicellular organisms or even in vitro systems and try and study these problems, right? So essentially, that is what I call the conservation of mechanisms, which allows us to develop model systems in biology, which helps us answer complex problems in laboratory systems. Now, since the question is, so if we are all that related, then how is it that humans are different, right? So that is one question that all of us keep wondering about. So, and this is a classical sort of image that you must have seen, even in your school textbooks, and even in your 12th standard textbooks, you'll see this classical image that is always portrayed, and which is that of Darwinian evolution. So we know in Darwinian evolution that we are all related to the the so-called missing link from where we all diverged as a family of apes. So the question is, what makes us different from the rest of the animals or rest of the plant species, rest of the cellular systems in nature, right? And that understanding comes from this, that what we have been able to achieve is language, right? We have been able to build a very complex language. And that language, what does it allow us to do? How does it make us different? Is that this language allows us to pass communication or information via non-coded genetic means, which is a soft memory, right? And then this also allows us the ability to transcribe that, to write that into books, to write that into computer programs. It helps us to transcribe that and learn from that into a hard format, right? So the books are the hard format, hard drives are your hard format. And then the additional thing is that because we can transition between soft and hard formats, we can do complex programming, right? So digital input can be converted into programmable inputs. So where is this relevant in today's talk? Because if you think about it, what this allows us to do is transcend the rate of genetic evolution. Because if I have to depend on only the genetic information in my cells, then I will have to go by only what DNA tells me. Whereas if I'm able to generate a language in my DNA, which can be encoded in soft means and can be programmed, then I layer on top of DNA and I transcend that rate of genetic information transfer that epigenetics will allow. Okay. And so essentially, I consider language as a form of transgenerational memory. And if you think about it, this is what the classical textbook image that we see of both DNA and chromosome. Today, I'm going to emphasize upon you how different these structures actually are. Okay. And what we know about DNA is that DNA information is also digital, just like a computer, which is made up of zeros and ones. DNA is also digital. It is made up of either an AT base pair or a GC base pair. So DNA information is pretty digital in that sense. The sequence is pretty digital in that sense. The genetic code, which is its letters, which make up this language. And then you have the gene, which is the unit of information, which are the words, right? And these words are then strung together into a language. But where is the syntax? Where is the grammar? Okay, how do I know how to turn these words into language, in what order to make sense, right? And when you try to understand that, that brings us to what we call as genome language. But before I get into that understanding of genome language, let me give you a quick video over how, well, I'll come to the video later, but let me first explain where this genome language is important. The central dogma. Remember the central dogma that I was talking to you about? The central dogma, as you can see, this is pretty familiar to most biology students, will know about this already. But what it allows us to do, the central dogma, where you take DNA information, transcribe to RNA, and translate to protein, what it allows us to do is that it separates the genetic hardware from the functional proteins, right? And then there's some functional RNA as well. But it essentially separates the function from the coding. And what this allows us to do is to keep the genetic information in a very preserved manner and pass it on very faithfully from generation to generation, while all the hard work that actually causes a lot of chemical problems and the chemical reactions that we go through in life can be done through these other machineries that include RNA and protein, right? So essentially, what are the features that central dogma allows us is stability of nuclear genetic material. It allows heritability of genetic material. It allows directionality of information transfer, which is what is depicted in this diagram. And then, most important, to which many people fail to appreciate, is that the central dogma allows amplification of signal. And that is very important for today's talk. Amplification of signal in the sense that one gene can give rise to multiple RNA copies, and multiple RNA copies, each RNA copy can give rise to multiple proteins. And different genes are amplified differently depending on the RNA and depending on the protein, right? So this itself, there is already, as you can see, there's already a type of amplification, and all these steps are part of the genome language, okay? So then where is the problem? So then what more is there to study? The important part that epigenetics brings to the table is at this step, at the first step of the central dogma, right when the genetic information is being read. Okay, so the obstruction of this genetic, if I can somehow stop the information from being read, what I'm able to successfully do is obstruct this path and abrogate the flow of information, right? Essentially, without creating a mutation, without altering the genetic structure, if I just block this step from happening, then keeping the genetic code identical, I can still change the type of information that I'm transmitting, or the words of the genome language can be changed. I can delete some words or I can add some words depending on what gene I turn on or I turn off, right? So let's talk about this regulation now. So what is this regulation in terms of blocking? So I'm calling it DNA accessibility, okay? So if you think about it, let's go through this quick video that talks
about 30 nucleotides upstream of the transcription region is the promoter, the core regulatory sequence of the gene. Next to the promoter lies the promoter proximal element. Promoter proximal elements stimulate transcription through the binding of tissue-specific regulatory transcription factors. Enhancers are regulatory sequences that may lie thousands of nucleotides from the transcription start site. Tissue-specific regulatory transcription factors bind to enhancers to stimulate transcription. When regulatory transcription factors are bound to DNA, they interact with other transcription factors. These basal transcription factors bind to promoters. They are required for transcription to occur, but they are not cell-specific and do not provide much in the way of regulation. When all of the basal transcription factors are bound, they form a basal transcription complex. One of the basal transcription factors, called a TATA binding protein, TBP, binds to a specific sequence known as a TATA box within the promoter. The TATA binding protein allows RNA polymerase to contact DNA. The basal transcription complex then recruits RNA polymerase and transcription begins. In cell types lacking the tissue-specific regulatory transcription factors, this particular gene would not be transcribed.
So there you have it, right? This is classical biology that we read in textbooks. Now, this is where I want to focus. This RNA polymerase that comes and initiates transcription exists in 3 different forms, which many of you who have done second year BSc level biology and molecular biology might know this already. And that is that RNA polymerase has a couple of modifications that can happen on RNA polymerase, which defines its functional state. Okay, there are 2 serines on the C-terminal domain of RNA polymerase, tail, and there are 2 serines. One is serine 2 and one is serine 5. So when both these serines are not phosphorylated, this type of polymerase just stays at the promoter and does not transcribe. So it's like the runner at the beginning of a race, which is ready to start, but is waiting to hear that gunshot, right? And then there is the serine 5 phosphorylated promoter, which is the initiator polymerase, which means it has heard the gunshot and it has triggered and has just started to run. And then you have the serine 2 phosphorylated polymerase as well, which is your elongation polymerase, which means that now it has heard the gunshot and it's already sprinting way across the field. So these 3 types of polymerase essentially dictate what type of transcription status your genes might be in. But this is transcription, right? So where do I differentiate this from epigenetics? How do I make this different from epigenetics? So that is what is today's sort of talk. So DNA, first of all, in this diagram that very nicely explained to you, assumed this whole thing by using naked DNA. And DNA is almost never naked inside the cell, right? And the second problem is that the obstruction, because DNA is never naked, so where are they? They are always bound to structures that are called nucleosomes, which we'll see in the following slides. But the binding of nucleosomes is generally obstructive to any DNA metabolic process, okay? That is kind of another fundamental that you need to keep in mind. So how does epigenetics achieve this control? So we are getting into the epigenetic mechanisms now. So what does epigenetics do? What kind of genome language it is creating, right? So here is an example. Here is a lady who has been just robbed and the thief is running away and says, help, help a thief, help a thief. And this police comes along and she's very happy. The police is actually letting the thief in, and she's and in fact, you know, abrogating the and helping the process. And she's asking, why aren't you arresting him? And the answer, sure enough, is commas, lady, because if you see, she said, help a thief. So what the police is doing is essentially helping the thief. So without that comma, essentially what you've done is you've altered the entire message. The punctuation is the essential point. So punctuation is what DNA accessibility essentially allows. And what epigenetics tells us is that there are various levels of punctuation. So this classical DNA structure that you see and the classical X chromosome, X-shaped chromosome that you see in your textbooks has many things in between which we don't see in textbooks. And that are these structures, that is, the DNA gets bound to nucleosomes, and nucleosomes are then ordered in several structures. Each structure is more and more compact than the previous one. And so to be able to access genes, you have to be able to open up the DNA structure and read them, like the previous video said. But if you don't want it to be read, it has to be packed away and stored away tightly, right? And particularly packed even more tighter the moment you want it to reach into a metaphase chromosome, which is the tightest form of chromosome, almost crystalline form of DNA packing. Okay, so this is essentially the packaging is essentially what is the epigenetic code. Okay, and we'll understand how this packaging, what are the different types of epigenetic codes that exist that alter these different states of packaging. So what do we know so far? So far, there is really, really strong evidence coming in from this end of DNA that there are histone modifications in DNA modifications. There are post-translational modifications on histones and modifications on DNA, both which alter the nucleosome biochemistry that allows for gene transcription. And then there is what is known as the solenoid model, which you'll see in the next video that I'm going to show. This packs into what is known as the solenoid fiber, which then packs further into these loops. These blue things that you're seeing are loops that are scaffolded onto the green matrix, and this leads to the chromosome. So, but the evidence here is really weak. Okay, this part is mostly by modeling. There is very little actual evidence there. The evidence that comes from the other side is the classical evidence of biology, which is karyotyping. Then people who are familiar with fish, which is in situ hybridization. And then there are these things help us define what are known as chromosome territories in nuclei. These are actually, if you look at the molecular detail, they are further divided into what are known as topologically associated domains. So these loops that I was talking to you about, each loop is attached to this green string at the base, the green matrix at the base, and therefore each loop becomes topologically isolated. It's like imagining that the DNA, the human DNA, which is actually a linear form, is now subdivided into several small circular forms as far as topology is concerned. Okay, so you can almost imagine that each loop behaves like an individual plasmid, right? Not really, but yeah, that's the equivalence I'm drawing to, that each loop can behave as a separate domain, topologically isolated domain. Okay, so, and where this is important and how do these punctuations, what do they achieve in biology? Let's look at that. So this is one classic example, because this is where epigenetics was first sort of discovered in the 70s and 80s. This was in Drosophila. Okay, what we see in Drosophila is that there are these genes that are linearly arranged on the chromosome, but these genes, even though they're linearly arranged on the chromosome, they fire linearly as well. That is, they're expressed linearly as well in a pattern, in a very segmental pattern. And this is what you see in the Drosophila, that you can see that they're color coded. So the front genes are blue, and then there's the purple genes and the orange genes and the red genes and the yellow genes. So you can see that they are going to fire in a particular order based on the segment in which they are firing, right? And this is extremely conserved even all the way to humans, not just in humans, even across in multiple systems of biology. So this seems like a very ancient program, right? Because all of us have this system. It seems like it must be a really ancient program that was encoded in the first common ancestor from where we all evolved, the eukaryotic kingdom, so to speak, the multicellular eukaryotic kingdom. So let's understand that further. So what we know is that, for example, during the introduction, you heard that from the same cell, which has the identical genetic material, we make up a whole human body, right? Where each cell has the same genetic material, the same hardware. And yet the kidney cell is very different from a heart cell, is very different from a brain cell, and so on and so forth, right? So how is it that we have the same DNA or the same hardware, but we are producing so many different games and programs that we are talking about? How are we able to achieve this? And that is what is epigenetic regulation. So essentially, if you look at the title that I've added here, the importance of syntax and grammar, which is the punctuation, is nothing but directly translates to the mechanism of syntax and grammar in genome language, which is packaging. Okay, so that is what I wanted to emphasize. So let's understand this a little further. And what is so perplexing about this packaging problem? Why I keep harping about this packaging? What is important to understand is that the human DNA, if you think about one cell, which contains 23 pairs, which is about 46 chromosomes, if you take the entire length of these 46 chromosomes, okay, these are 6 billion base pairs, which is 3 billion times 2. And if you take these, and we know that each base pair has a rise of about 0.34 nanometers, right? Now, if you multiply this rise into the entire 6 billion base pairs, what you get, a total length of DNA, which is about 2 meters long. So essentially 2 meters of this molecule is packaged into our cell, into our nucleus, which is only a few micrometers across, right? So this is a huge problem, huge compaction problem that we're thinking about. And just to give you a fair idea about what this actually means is that let's think in terms of Maggi noodles, right? Maggi noodles is about 2 millimeter diameter, average noodle. And if you take the length of the noodle, then that human DNA from a noodle from one cell, that extends from Mumbai to Chennai, right? And that needs to package into your average classroom that you sit and attend your classes in. So this, you can see, is a big problem, because you want the DNA to be packaged. At the same time, you want the information to be available to you to read, to do your whole biology. Otherwise, we would only be rocks, right? So how is this million-fold compaction achieved? And how can such highly compacted DNA regulate varied patterns of gene expression accurately? So let's listen to this about how the DNA is packaged.
DNA is like a long string composed of a double-stranded helix, folded into very compact structures in the cell nucleus. The center of each of these structures is composed of a protein called histone. Each center consists of 8 proteins, 2 copies each of 4 types of histones. Researchers have been working hard to understand their roles. This structural unit, formed by DNA wrapped around the histone proteins, is called a nucleosome. Human DNA measures about 2 meters in length. But it can fit in a tiny nucleus of only 10 micrometers in diameter by forming a fiber of packed nucleosomes called chromatin.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin.
It is also known that changes in histone state can alter DNA fluctuation. Histones have unstructured, flexible regions. These are called histone tails. When the histone tails undergo chemical modifications, such as acetylation, the structure of the nucleosome changes. For instance, we know that acetylation of specific parts of the histone tails loosens the chromatin structure, making the nucleosomes less tightly packed.
This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes.
Right, so let's recap quickly because we have already seen all of this. So we know that the nucleosome is made up of 8 proteins, which is what's called the histone octamer. These histones are made up of 4 types, and these are your H2, H2B, H3, H4. An octamer is what binds to 2 dimers, H3, H4 tetramer binds to 2 dimers and makes a histone octamer, and that is what wraps the DNA. So let's understand now what this does. So this is the actual crystal structure of the space-filled model of the nucleosome structure. You can see that it's pretty tightly packed, right? And what this achieves, essentially, if I now schematically depict this nucleosome into a penciled diagram, you will notice that the promoter, essentially the accessibility of the promoter, which is what RNA polymerase binds, is important because based on where the promoter is packaged and where the Tata binding sites are packaged, the RNA polymerase accordingly will have that much efficiency to transcribe that gene. So if this promoter and the transcription factor binding sites are part of the nucleosome complex, then that is poorly transcribed or not transcribed at all. So it is inhibitory to transcription. Whereas if this part is now remodeled, so if you can imagine these 2 structures as knobs, as like a regulator, right? So you can turn these knobs and regulate the gene expression. You can, by turning the knobs either ways, you can therefore expose more DNA or release the transcription factor binding sites, and thereby then allow the transcription factors to bind. So there is a hierarchy in this process. Some transcription factors require this DNA to be opened when they can bind, and some transcription factors can override this nucleosomal capacity, right? So that's a subtle difference which some genes have. But most genes are obstructed by binding of nucleosome to these transcription factor binding sites. So essentially, the chromatin remodeling, which is this part where I move nucleosomes around on the linear piece of DNA, this regulation that I talked about is essentially your remodeling process, which changes where the nucleosome sits. That is called the nucleosome position. And in a population of cells, say I take a clonal population of 100 cells, how often will I find that nucleosome present at that exact same position? That is known as occupancy. So nucleosome positioning and occupancy is what will decide the rate of gene expression, right? So this is your first hallmark of epigenetics, that nucleosome is the basic unit of chromatin and is generally inhibitory to transcription. Okay, and so how are nucleosomes modulated? Obviously, this tuning that I'm talking about, how is this tuning modulated? So let's think about it. Now, let's look at the DNA a little more from its chemistry and from what makes it up, right? We know that DNA is made up of this phosphate backbone, which is a huge number of phosphates. So it's really a highly negatively charged molecule. So if you want to compact your DNA, these phosphates are going to repel each other, and the DNA it will not be able to get compacted because it's so highly negatively charged. So therefore, it is important that this charge is somehow undone, and what undoes that charge is the positive charge of the histones. So histones are highly basic proteins which are highly positively charged because of the arginines, lysines. They are rich in these amino acids, and that's what neutralizes some of this charge and allows the DNA to package, right? Not only neutralizes, because opposites attract, therefore this binding of the nucleosome to DNA is very strong. Okay, the unique thing that you probably see, even in the video you saw, these tails that they talked about, which are unstructured, are actually unique because the density of these positive charges on these tails is much higher. Okay, so these tails, you can imagine them like if my palm is the nucleosome, you can imagine that in this palm, if this is the DNA that is wrapped, these tails, these are histone tails that are unstructured. If they are negatively charged, then if they are positively charged, then these tails will strongly bind to DNA over the DNA that is already wound, thereby making it really tight. It's locking it in, right? Whereas if I now add acetyl marks or negative charge to these tails, then I'm neutralizing those charges and I'll release these tails from binding to the DNA, right? And that is exactly what the first modification of DNA that I'm going to talk about achieves, which is basically the histone acetyl modifications. So all histone acetyl modifications have the same mechanism. Okay, then comes the methylation, the tail lysine methylation. So the tail is very important because it has a lot of modifications which are part of this unstructured region of the tails. There are many modifications that happen in the core as well, but today I'm going to talk a little bit more about the tail modifications. Okay, so the tail modifications largely comprise of methylations, but it is not like acetylation that it has only one phenotype. Okay, that it can activate genes. So if I acetylate histones, the gene gets activated because the DNA is freed. But if I methylate, there are two possibilities. One, depending on where it gets methylated, if the K4 or the lysine on the histone H3, the fourth lysine on the histone H3 gets dimethylated or trimethylated, then that gene is supposed to get activated. Whereas if the same lysine on a different position, say the lysine in the 9th position or the 27th position gets dimethylated or trimethylated, so the chemistry is same, but the position matters, the context matters, these nucleosomes will be mostly there in suppressed or repressed genes. Okay, so then there is the third layer of DNA methylation. Now, as you can see, the histones are non-covalently bound, right? And the DNA is where the genes actually are being able to be transcribed. So the DNA methylation is the kind of the closest covalent modification that you can get that will go along with the epigenetic phenotype. And DNA methylation happens on two different types of methylations. I'll come to that, but essentially in humans, it is present at what known as CpG dinucleotides, okay, which means that in a genome you can have C in any context. The next nucleotide can be any other nucleotide, but whenever a nucleotide is a CpG, which is C followed by a phosphate followed by a G, that dinucleotide, CpG dinucleotide, is often methylated. Okay, and we'll come to that as to why that happens. And this methylation leads to gene silencing. Okay, so it's also a repressive mark. So as you can see, these set of marks on the histones are non-covalently linked to the DNA, whereas the DNA methylation is a covalent link. Okay, and this is important to appreciate because this has very good implications in mechanism. Okay, and we'll come to that in a little while. So the mechanisms essentially can be looked at in two ways. One is, of course, the function as to how this leads to gene expression. And the second one is inheritance, because remember, we talked about this being transgenerationally inherited, not only through cellular generations. All right, so now this slide is a little complicated, but I'll go through it quickly. Essentially, it's showing you a number of different modifications that are possible on histones. There are close to about 65 different modifications that are possible. And then these are the combinatorial effect of these is what can affect gene expression. Not only modifications of histones, there are also something that are called as histone variants. Okay, histone variants is where you have an alternate type of histone that can get incorporated and change the biochemistry of this nucleosome, so that now this binding is different from this binding. Okay, of DNA interaction is different. Similarly, there is what is known as nucleosomal interactions based on these histone modifications. And the last is what creates these differences is essentially the chromatin remodelers, which either compact them or space the nucleosomes out by releasing the DNA. Okay, so how do histone modifications influence? So these are all about histone modifications, which are all the non-covalent bonds, right? Question is, remember in the previous slide, I said that there is this two different types. One is covalent and non-covalent type of interactions. See, remember that these are all covalent modifications as far as the histone is concerned, but these are non-covalently attached to the DNA. Whereas this modification, the DNA methylation, is covalently added directly to the DNA. Can this type of modification influence this type of modification? Okay, and the answer to that is in the next slide, which is essentially yes. So what is known is that the K9 type of trimethylation drives DNA methylation, and this means that there is an epistasis in relationship. Just like you have heard of epistasis of genes in glycolysis, for example, where you have gene A. If A gene gives rise to B gene, the gene product interacts with gene B. Then if you knock out A, then B will not be able to do anything, right? So A becomes epistatic to B. Similarly, here we suggest that the K9 modification is epistatic to the DNA methylation. So if you knock down or knock out K9 modification, then you can prevent DNA methylation from coming in. And why this is important, we'll come into the next slide. Okay, but essentially, I am going to rush through this now because there are so many different modifications that I have told you in the first few slides, and then I have listed them here. There's so many different modifications of epigenetics that can be clubbed together to create a gene transcriptional state. So how are they all related, right? There's no time for me to go through each one of them. So how are they all related? That's a lot of mechanisms. How do you connect them? So this is sort of where I bring my second hallmark of epigenetics, which is essentially, you think about epigenetics as a fractal spreading of the same state. Okay, let me elaborate how that happens. So essentially, this is how one of the first phenotypes of epigenetics was identified as position effect variegation, again in the organism Drosophila. So what is known is that DNA methylation can be of two types, either cytosine methylation or adenosine methylation in eukaryotes. And we talked about CpG islands, right? So let's look at CpG as an example. How is CpG, why is CpG being methylated? Why are not other Cs being methylated, right? So if you think about it, when the DNA is being replicated, so these are the two daughter strands that are being replicated, this is a CpG. Okay, this CpG has, if you think about it, on the opposite strand is also a CpG. Right? So it has an exact mirror image on the opposite strand. So both Cs can get methylated. Now when this DNA replicates, you have semi-conservative passing of information. So the CpG, one methylation will go to one strand and the other methylation will go to the other strand. Right? Therefore, that results in what is known as a hemi-methylated state of DNA. And what enzymes, DNA methyltransferases can do is that they can recognize these hemi-methylated states and reset them back to the completely methylated state. So you can see that this allows, the CpG dinucleotide structure, therefore, allows you to have a very faithful replication of the epigenetic mark, right? So if once a gene is shut down, it can remain permanently shut down. So a CpG methylation is therefore, DNA methylation in general is a very strong mark, okay? Now let's contrast that with the other types of modifications, which are your histone modifications. So what we know is that histone modifications are of, again, can happen because the histone proteins are so many proteins, it can happen on one and not the other. There are always pairs of histones. There is an H3, two H3s in the same nucleosome. So one H3 can get modified differently and the other H3 can modify differently. So that can, this entire complex this can create a complicated set of outputs, right? So that is what I'm trying to give in this type, simplify it. It can be cis or trans modified peptides, which I'm not going into details into, but that's the essence of this understanding. And then there are histone variants, which I told you that the whole histone can be swapped for a new histone, right? So because there are two H3s, if I swap one of the H3s away to a new H3, that is a different type of H3, then now what I generate is what is known as a heterotopic nucleosome. Whereas if both the H3s are identical, then what I have is a homotypic nucleosome, right? So based on both the types of histone variants as well as the marks, you can generate so many combinations, right? So if you do a mathematical sort of calculation, this can lead to a huge number of combinations. Okay. Now these three marks all together, these combinatorial effects, is essentially what leads to which tells the chromatin remodelers which way to go, either to loosen up the chromatin or package the chromatin further, right? By testing its positional, by testing where the nucleosome is sitting on the gene. And another pathway which I haven't really talked about is the emerging involvement of non-coding RNA in these epigenetic processes. Okay, all your siRNA and so on and so forth. They can also interact with the epigenetic processes, long non-coding RNA particularly. And these also interact with these same three mechanisms. The non-coding RNA also interacts with these mechanisms to finally give rise to what is known as DNA packaging and gene expression. So this is your summary of epigenetics in a nutshell. Okay, so I've explained to you about how the function is. Okay, I've explained to you DNA methylation inheritance, how that can happen. Let's talk about how nucleosome modifications, the histone modifications, how can they be inherited, right? So now let's simplify this. To simplify this, I've just drawn some structures here. So this is that 8-octamer histone, right? I've just colored to give you an idea, say about how they will replicate. Say, for example, these two are marked as Z, which are two variants, okay, H2A variants. So Z is an H2A variant. So now let's think about how this nucleosome will replicate. So when this nucleosome can replicate, it can immediately replicate into two if the enzyme, as the DNA is replicating, the enzyme comes and replaces this and adds this new histone there, right? Or the mark. The other way it can happen is that this old nucleosome can get transferred to either one of the new daughters, and therefore the other daughter gets a completely brand new nucleosome, right? And then eventually, as the cell cycle proceeds, that gets reset again by chromatin writing machinery. The other model is that because there are two copies, it can get semi-conservatively split into both the nucleosomes, and therefore both daughters get half of the information, and that allows, just like CpG methylation, this allows the complete resetting of the original mark, right? And the third model is that we can completely evict this nucleosome during, you can actually lose the nucleosome during transcription or replication, and once the nucleosome is lost, then obviously something has to come in there. It will be a new nucleosome, and that new nucleosome eventually has to get reset into the mark, right? So out of these models, what we know for sure is that this model does not happen. The semi-conservative model does not happen. Nucleosomes do not break apart into equal halves and go into two equal daughters. And this is very important, okay, because this gives a distinction between mechanism of how DNA methylation is transmitted, how that information is transmitted, versus histone modification information is transmitted. And what this tells you, therefore, that this can introduce variability in gene expression, right? Because sometimes you don't end up with the original histone, sometimes you end up with the original mark, and so you reset the original mark. But sometimes you lose that, and you end up with something altered state of gene expression, right? So this is where the environment can start influencing your epigenetics. So let's look at this a little more closely. So this is the third hallmark of epigenetics, which I state is that epigenetics can spread, okay? So this is the third hallmark of epigenetics, and yes, the inheritability is linked to the spreading, the second hallmark, which was spreading. So let's look at this a little bit closely. So here I have drawn a few nucleosomes that are all, so to simplify the complicated figure, I'm going to highlight only that, right? So I'm going to highlight this where you can see these red and blue dots. These are the modifications. So each nucleosome has two sets of modifications, okay? What we can see is that this enzyme that binds, once it replicates, what happens? So let's look at there's an enzyme here that can recognize the red dot on one nucleosome, but now put that red dot on the next nucleosome, right? So it replicates that same mark on the next nucleosome, not the original nucleosome, right? And there's another enzyme, for example, this enzyme, that can recognize one mark and put the other mark on the same nucleosome, right? So these are the different types of mechanisms that exist. And what I've shown you exactly in this figure is an enzyme called Polycomb repressive complex. Okay, so this is a complex that does and resets these marks in that way. So if basically, even if you get a heterogeneous segregation of nucleosomes between the two daughters, because the marks can spread, because the marks can reinforce each other, therefore the original state is overall maintained in a more faithful manner. Okay, so this is the protein that does it. And how does this protein do it? If you think about so this is actually 3 proteins put together in a crystal structure. And if, again, this is very complicated for everybody to look at, so let me simplify this image. I will blacken everything out, and I'll highlight where the active site of this protein lies. So this is where the active site lies. Okay, so you can see this green peptide. This green peptide is what the histone peptide is, which can get trimethylated by the Polycomb complex. And now, if you look at the protein more closely, you see that there is another part of the protein that also has this green peptide binding site. So the same peptide, the trimethylated histone peptide, can bind at 2 sites on the protein. One is the active site, and the other site is the allosteric site, which influences the activity of the active site. Right, the same mechanism that we talked about in the original in the previous figure. All right. So kind of towards the end of the talk, so I'm kind of going to summarize a few things before I end the talk. And so the summary is that you have different states of gene expression that can be that you can kind of create, right? You have silenced or repressed genes, and then there are poised genes, and then there are stem genes. These poised genes are very important in stem cells, and I'll talk to you about that in the next slide as well. So essentially, if you compare these gene states, they differ in DNA methylation, right? So they differ in the states of DNA methylation, right? So you can see that in my diagram, I have put these black lollipops, which are essentially a CpG methylation. And as you go into express genes, the promoters get less and less methylated, okay? And this is what was called in epigenetic biology as heterochromatin, which means shut down chromatin, or chromatin that is completely packaged, tightly packaged, okay? And this can exist in 2 forms. One is the constitutive heterochromatin, which is like your X chromosome inactivation. And then you have facultative chromatin, which can actually shuttle back and forth between active and repressed states, okay? And these 2 chromatins are, as the name suggests, there can be marked with different histone modifications, okay? And then you have the next pattern of gene expression. As the nucleosomes are remodeled, you can see that the nucleosomes are more spaced out, the promoter is exposed, and the gene can get expressed, or the nucleosome can be even kicked out and lead to nucleosome eviction and lead to very high active genes. And this is what is known as euchromatin. So this end of spectrum of chromatin is called euchromatin. But these, as you can see, are not binary features as we try to think of them. These are there is a lot of shades of gray in between. Okay, and this euchromatin is also marked by specific chromatin marks. So where is all of this relevant? So what we know about cells is that cells differentiate from stem cells into a differentiated state as we grow, right? So we start from the zygote, and we eventually are a bunch of differentiated cells and stem cells. And all of these activities play a role there. And the 2 Nobel Prizes that were given in the last 20 years were based on these activities as well. Both Dolly and IPS were very closely linked to epigenetic understandings as well. Okay, so what Dolly achieved was that it took a differentiated nucleus and was able to de-differentiate it. Essentially, take these genes which have already differentiated into its or committed into its cell types, either expressed or not expressed. Basically, if you think about it like a skin cell, right? A skin cell will express all the genes that are required for a skin cell and will maintain it at an active state. But all the genes for a kidney cell in that cell will be suppressed or repressed, right? It can keep it completely packed and therefore not access that information at all. And so the differentiated cells can actually segregate this information in cells like that. But what Dolly showed is that we can rewire this information back to the original state, which is called as bivalent chromatin, which means that both the opposing marks and the repressive marks are now there on the same region of DNA or the same region of chromatin. And this bivalent state is what people think to be the epigenetic nature of stemness. Okay, so if you think about this now in a more sort of cellular manner, what this allows you to think about is if you think about a ball rolling down a hill, this ball can end up in several valleys. Okay, so this is the original stem cell, the totipotent cell or the pluripotent stem cell can end up differentiate into different lineages. Okay, but what we have seen that as people age, sometimes cancer, the cells de-differentiate or go into what is known as an EMT transition, de-differentiate and become into a cancerous cell. What our studies have shown, so studies that we have been involved in the last 10, 15 years, is that this cancer cell, which can form as a result of de-differentiation or EMT transition, can also happen due to environmental stress. Okay, and the biggest environmental stress that we have discovered is ROS-mediated stress on these bivalent promoters. And what happens is that this changes those stem cells that are there in your body, and that stem cell maintains that now an altered information of epigenetics. That altered information is carried on forward and probably becomes the tumor initiating stem cell that eventually gives rise to tumors or multiple different types of cancers. Okay, so these are the two understandings of how epigenetics can give rise to cancer. Of course, these are things that are still being actively worked upon, and there's a lot of active research going there. So therefore, cancer essentially is nothing but a developmental program gone awry, right? And so the question, therefore, can the environment, can what we eat or what we do directly affect our epigenome? So one last example I want to show you before I end this talk is that what we eat does affect our epigenome, because everything that I showed you, the methylation and the acetylation, the two main characters, right? You look at this is the methyl donor. The methionine comes, the SAM, which is the methyl donor, is actually coming from methionine, right? And then there's the acetyl coenzyme A, which is your acetyl donor. Both of these marks are what regulates epigenome. So depending on where these marks are coming from, what is the balance of these marks, balance of these molecules in your gene, in your cell, in your cytoplasm, that's what eventually affects the epigenome as well, right? So there is a strong link between metabolism and the epigenome and how the epigenome is maintained. And this is the classic sort of one example that I love giving is a quick story that I'll tell you. There were two identical twins. If you analyze the DNA methylation status of two identical twins, okay, and say they grew together. So until early childhood, most twins are always staying together. And this is a story similarly like that. This is DNA methylation profile of different chromosomes from two identical twins who have stayed together. 3-year-old twins have until 3 years they were together, right? So there is not much change if you compare their chromosomes. This chromosome is from one twin, this is from the other twin. These are genetically identical twins, okay? But there was this case where the two ladies got separated, two children got separated during the Nazi times, and one went to Europe and one remained in Europe and settled down with their family in Europe, while the other twin moved to America and ended up with different lifestyles. And no marks for guessing, but sure enough, because they had different lifestyles, they had differences in their disease susceptibilities, like I told you before. And so obviously one remained healthy and one ended up with cancer. Okay, so then they analyzed the same DNA methylation profile for these two twins. Look what they found. These boxes highlight those regions that you can see are clearly different in the twin that ended up with cancer. The DNA methylation profile is quite different from that of the twin that did not have cancer. And these are gross methylation changes that you can see even in chromosome karyotyping, right? By FISH or by DNA methylation hybridization. So essentially this tells you that epigenetic changes do occur due to environment. But can this result in mutations? Can this become permanent? Can these changes become permanent changes in our genome? And the answer to that is that maybe yes. Question is how? And how comes from our understanding of basic genetics, right? So what is genetics like? Here is an example of cytosine. I've drawn these structures to explain to you better. So you can look at this, the thymine and uracil are different in this methyl group here. And cytosine and thymine are different in this amine group here, right? So now you can imagine that if I transfer this methyl group here, I get a cytosine that is methylated at the 5th position, which is nothing but 5-methyl cytosine, the same cytosine methylation that I've been talking about so far. And what is known in biology is that these methylated cytosines can get oxidatively deaminated and turn into thymines, right, or uracils for that matter. And this process of oxidative deamination of methylated cytosines, the DNA that gets methylated, can lead to an C to T transition or a mutation, right? So, and this is some data to quickly show you that yes, if you look at certain colorectal cancers, there is a direct correlation between the mutation rate per MB of this particular colorectal cancer and the amount of CpG method that gets methylated. There's a direct correlation of the methylation with the mutation rate. So this kind of points to the idea that methylation can drive mutation, right? So to summarize everything that I've told you, that we are essentially who we descend from, right? Our genes tell us who we are. We are also what we eat and how we live, how we choose to live in our lifetime. And then we can also pass that information on, because we can pass it to our children, to our grandchildren, right? So question therefore is that, was Lamarck right after all? Because Lamarck said that the environment can dictate how the giraffe's long neck can get selected for. And here now we have this new emerging field of epigenetics that is trying to understand these effects from a Darwinian point of view, right? How environment can change us as genetic beings during our own lifetime, and in a heritable manner that we can pass on to our generations, and that can actually transcend the rates of genetic evolution. With that, I would like to sort of end my talk with the definition of epigenetics, which is heritable changes in gene expression rooted in chromatin mechanisms without changes in the primary DNA sequence. That is the definition of epigenetics. That will be differentiated from transcription with any other type of heritable biology that we know about. So this is the special definition of epigenetics. Okay, and it of course has normal underpinnings in development, imprinting, differentiation, and cell renewal patterns and all that. So essentially it is all about how our DNA is packaged. And if you think about diseases, non-communicable diseases, not just cancer, cancer is just one example, but schizophrenia, you name it. There are so many non-communicable diseases. And these non-communicable diseases are essentially the abnormal way in which our cancer cell DNA is packaged. Essentially, that is what will define as the cancer epigenome, and how that is inheritable, leads to inheritable patterns of gene expression, which foster tumor initiation and progression. That is how we define the cancer epigenome. Okay, so let's, with this understanding, I hope today you have a better understanding of what epigenetics is, what is the molecular mechanism of epigenetics. And so let's take this. I implore students to come and interact with us, come and visit our labs, and even study more about epigenetics and ask more questions. Look at, there are hundreds of talks on epigenetics available online. So please go ahead and educate yourself about this very new field of epigenetics, which is really going to be the next big push in how we treat diseases, how we personalize medicine to each individual. Thank you.
Thank you, Dr. Subhajit Sen. And your talk was amazing, I should say. And we're really thankful to you that you gave them first-hand information about epigenetics. And you're also inviting students to have a look at your lab. And I'm sure they're going to do a lot of progress, and we thank you for that. So thank you, Dr. Subhajit Sen, for that thought-provoking talk on epigenetics, which was essentially an excellent blend of basics and in-depth discussion. You really made it so simplified for us to understand. So the audience has been buzzing with questions. I call upon Mr. Suman Gangar to conduct the question and answer session. Over to you, Suman.
Thank you, Shweta ma'am. Dr. Sen, a wonderful presentation. And actually, our WhatsApp group is getting exploded with the questions. We won't be able to ask all, but here are some of the questions which have been moderated. And the first one is, though you mentioned about how the lifestyle is going to have an effect, but one question is asked, how does diet affect epigenetics or neurological disorders? Maybe
so yeah, so that's a very good question, because we study in our textbooks of this thing called the Wiseman Barrier, right? Wiseman Barrier essentially is that your somatic cells, whatever they go through, if you're stressing somatic cells, that information can never go to your germ cells, because somatic cells are not talking to your germ cells. So how is it? So that's what was called classically the Wiseman Barrier, that genetic information can get passed on, and you can probably end up with cancer, or whatever you do in your lifetime does not affect the genetics or the epigenetics of the later this. But what we've learned from epigenetics is that this is true, that there is a possibility that diet actually plays a role. Now, where does diet play a role? There are these epigenetic machines that I told you. I already showed one slide where both the methionine pathway as well as the acetyl coenzyme A both are linked. directly. So methionine is strongly linked to the nucleotide metabolism and the amino acid metabolism pathways. And site acetyl-CoA, I don't have to tell everybody, it's directly linked to the glycolysis and the TCA cycle, right? So both of these are the core metabolic pathways that ensure how we how our genes are going to be sort of made, new genes are going to be made, and what's the health of our cells. And the balance of these molecules will directly interact with the epigenetic molecules that put these marks on these genes or the epigenome. Okay. Now there's another layer. For example, NAD. NAD is a molecule that is involved in it's a redox molecule. So NAD is it exchanges between two states, NAD plus and NADH, right? And the reduced and the oxidized states, these reduced and oxidized states are what are part of the metabolic chain. But what we know is that there are histone acetyl transferases called, especially one group called sirtuins. These acetyl transferases actually interact with these with the NAD machinery by sensing this in the cytoplasm. These this sirtuin 1, which is normally present in large amounts in the cytoplasm, translocates into the nucleus, and then puts this sort of deacetylates some of those genes, right? And so how it goes and targets the genes, which gene it goes and does, those are details that will come about depending on what program you're looking for. And of course, we don't understand all the nitty-gritty details of this process. What we do know is that direct metabolic processes are involved directly with epigenetic processes, because all these common molecules are involved. And a good example of this is the diet-based example that you asked about, is that if I maintain a healthy diet, and what is known as what in epigenetics, there are studies that are conducted. For example, this NAD, the sirtuin 1 that I talked to you about, sirtuin is highly implicated in increasing lifespans of individuals. Okay, and this is proved in model organisms as well as mice. So it actually increases lifespan. If you knock SIRT1 down or you remove SIRT1, lifespan reduces. Right. So clearly SIRT1 has a major role to play in lifespan, in how long we live. And this has to do with how the epigenetic marks are placed on our genomes. The older we get, the epigenetics keeps changing as we age. And so we can actually classify if we study these epigenetics, we are now coming up with what is known as the epigenetic clock, right? So there are different, the same individual, you can look at the type of epigenetic marks that exist on a particular gene and observe that over a particular set of lifetime and see how that epigenetics is changing and call this the epigenetic clock. So if I change my lifestyle, we know now that if I change my eating habits, if I maintain good calorie restriction of my eating habits, this epigenetic clock slows down. Even though chronologically we keep aging, the epigenetic clock actually slows down. So diet has a direct relationship with your epigenetics, and that has a direct implication on how genes are expressed, which has a direct implication on how long you will live.
Wow. So I guess diet is not only just important for me, but even for my coming generations.
Exactly. That's what we are learning now, that this can be possibly transmitted to. We don't know how much of this is going to be transmitted, but we can see in controlled mice experiments, we do see this, that diet can influence the epigenetic signatures that can be transmitted.
Okay, we move on to the second question. Why is global methylation upregulated in cancer?
Actually, that is untrue. It is actually the promoter methylation that is upregulated in cancer. So can I use a slide to quickly show a quick example of what I mean?
Sure. Shashank, please help.
Yeah, I'll do the slide. I'll share the slide so I guess students can see that. Here we go. And I share that. Okay. So here is the answer to that. So essentially, this is the epigenome of a normal cell. What happens is this is the promoter, this is the transcription start site, and the normal promoter is usually more loosely packed than the rest of the genome. And so the rest of the genome is highly packed. And what is seen in these black lollipops are that the promoter is usually unmethylated compared to the rest of the genome, which is usually methylated. The CPGs, I'm talking the CPG methylation here. But what happens, what changes I'm going to go past these, what changes as a window of the environment is that in cancer, what we know, now these are two endpoints, a normal cell and comparing that with a cancer cell. Okay, when you look at the cancer epigenome, it is completely switched around. Many of these promoters, which are tumor suppressor genes particularly, get hypermethylated, and the rest of the genome loses large amounts of methylation. So the rest of the genome gets hypomethylated. Okay, so this is what is seen in cancer versus normal cells.
Hello, Subhajit.
Hi.
Yeah. So there is another question. Does lifestyle, essentially levels of stress and or meditation, affect the level of acetylation or methylation, thereby changing what, when, and how much is read for expression?
Right. So that's a very good question, because we are all constantly wondering how much our lifestyle contributes, like stress, yoga, or meditation, and even not just that, even exercise, for example, regular exercise, right? Now, what is seen in mice experiments, this of course we can't do in humans, because see, we have to always figure, whenever we are doing science, we have to always figure out what is correlation, what is coincidence, and what is causality, right? If two things happen always together, there is not enough information for us to tell that those two events, because they are always happening together, one results in the other, right? This is the sort of what modern science brings to the table. And this is what makes modern science different from so-called sort of classical thinking, right? Where we look at correlations a lot. Now, what if you think about this process that we just asked, exercise in controlled mice experiments, which is seen that if you prevent the mice from exercising, or rather just put it in a sedentary environment where it has nothing much to do, and compare that mouse with an environment which is really enriched, where the mice is continuously active, running around, or what we're doing things, running mazes, solving problems, then those mice tend to live longer or have better epigenetic clocks than compared to these mice which are leading a more sedentary lifestyle. So I would say that any form of exercise is good for you, whether it is yoga, whether it is this, or running around, or whether it is just doing Pilates or any form of exercise, just you dance around, just lead an active lifestyle. And just an active lifestyle is good enough for your body to maintain, keep your epigenetic clock constantly going. The moment you become sedentary, the moment you start altering your lifestyle habits, eating as well as the stress, take stress, go into depression, those kind of things will obviously lead to a sedentary lifestyle, and those will alter your epigenetic marks. Now, in human studies, we don't know what is the causality, because we can't impose these things on an individual and observe him before and after what happens. So most of the human studies draw correlations. So we don't know, we can't make the mice perform yoga or de-stress the mice by some, we can do this on mice, but we can't do this on humans. So it's very hard for me to make that jump unless we find a molecular correlate in mice, and then see if that molecular correlate is true in humans or not. Therefore, molecular biology is very important in all modern studies, rather than just assuming that eating, drinking, kadha se leke, any kind of traditional thing is going to be helpful for me. It is important to understand what is the molecular basis for those things, and understand whether it's actually affecting molecularly, or is it just a correlation. If you look at correlations, everybody who drinks water dies. So then can I then say that drinking water is harmful for us? Obviously not, right? So that's a stupid example, but that's a typical fallacy of correlations.
Right. So I guess everybody here is has listened to this, and active lifestyle is very, very important. Moving on to the fourth question, does epigenetic regulation occur in bacteria?
So the answer is, we know that regulation occurs. We don't call it epigenetic regulation in its strictest sense, because as I mentioned to you about the definition of epigenetics, is that it has to do with chromatin modifications. And if I define chromatin, it has to do with nucleosomes and how nucleosomes are made. So if you loosen up this definition, if you want to say that does it have anything to do with the matrix associated to DNA, then maybe you'll find something in bacteria. But the moment you start loosening definitions, you'll obviously start finding some mechanism that is related in one way or the other. It cannot be true. Having said that, it cannot be true that epigenetics suddenly appeared in eukaryotes, right? DNA methylation exists largely in prokaryotes as well. But in prokaryotes, it is used for a completely different mechanism. It is used to defend the cells from foreign DNA, from viral attack. So the cell's DNA, for example, E. coli will have EcoR1, and it will methylate all the DNA of the E. coli, of the EcoR1 sites. So the EcoR1 of its own cell will not be able to chew the EcoR1 site in its own DNA. The moment of new viral DNA comes, which is not EcoR1 methylated, that EcoR1 site can go and get attacked by the existing EcoR1 enzyme in the bacteria. So it was an immune mechanism. DNA methylation was an immune mechanism. But then it was hijacked slowly as we evolved into this epigenetic regulation, which is a tunable form of regulation. But let me tell you that in epigenetics, in eukaryotes, in mammalian systems, in humans, we still do use epigenetic regulation for immunity against viruses as well. Not in the classical sense of immunity, but you imagine that during evolution, we have collected a lot of viral baggage in our genomes. In fact, 40% of our genomes is actually viral DNA from previous infections that have integrated into our genomes through evolutionary time. Remember, 3.4 billion years, right? So we have accumulated this evolutionary baggage. And how do we keep that baggage completely silent all the time? By using DNA methylation. So we are still using DNA methylation to keep the viruses in our genomes, the latent viruses in our genomes, silent, right? But when new viruses come, those are different immune mechanisms. COVID-19 and all these things are different mechanisms to deal with. I think that's about it.
Thank you, sir. The next question is behavioral epigenetics heritable? Could you explain with an example?
I started the talk with that example of mice, which was fear conditioned. So if I want to explain that fear conditioning, what is fear conditioning? So these mice are normally not fearful of, say, smell of banana, for example, right? The mice don't get scared of smell of banana. But now what you can do is every time you give the mice a smell of banana, you will give a small shock to the mice, electric shock to the mice, right? So the mice obviously does not like the electric shock. It exhibits a fear response. It starts, the freely moving colony of mice, they will start huddling together, they'll start shivering, their tails will shake. So these are typical fear responses that you can see in mice, right? So now you can keep repeating this. So this is what is called as fear conditioning. So every time you give the smell of banana, you give that mice cage a small shock. So now the mice start to condition themselves that, oh, every time getting the smell of banana, I'm going to get a shock, which means that something is related there, but I don't like this banana smell anymore. Every time I smell banana, I'm going to get a shock. So now when you give the mice just the banana smell, these fear conditioned mice, now once you give it the banana smell, it will start showing the exact same fear conditioning responses that it showed even without giving the electric shock, right? So the fear conditioned mice maintain this phenotype even after the shock is taken away. Now just providing smell of banana will make the mice fearful. This is what is behavioral change, which is, of course, we think of it as a neurological change, right? Behavioral change. Fear is very neurological in our sense. But this was seen to be transmitted across generations, even to the F2 generation, which is the grandchildren. How it was transmitted, what were the mechanisms, is currently sort of looked at. But the clues lied in those papers, the clues came from DNA methylation. So that's about it for these kind of things. So yes, behavioral change, behavioral things, at least from the mice experiments, there's clues that we can gather. So is it true that if my grandmother was very afraid of cockroaches, maybe that's why I'm afraid of cockroaches? I don't know.
It's interesting.
Right.
Okay, next question is, there is now an evidence that methylation is related to cancers like colorectal cancer. How can we use this new perspective of epigenetics in treatments?
Right. So as I explained that the DNA methylation can lead to, so I haven't really talked about it, but DNA methylation is a silencing mechanism, right? So again, I'm going to use a slide quickly to sort of explain this. So it's just easier with slides. Okay, here we go. Sorry. So I mean, I don't think I can find that slide, but essentially, okay. Yeah, here. So I have to share the screen. Here you go. Okay, so what I've kind of trying to explaining in this slide is the sort of the first you understand the genetic basis of cancer, right? So cancer is a genetic disease we know very well. So how do you explain that? We know normal cells have two types of genes. One is the accelerator genes and one is the brakes genes. Accelerators are your oncogenes, and brakes are your tumor suppressor genes, right? So when you look at the central dogma, when these genes get mutated, these normal genes get mutated, and therefore that leads to altered function, and the brakes are lost, right? And due to loss of function of the brakes, there can be more mutations that can accumulate that can lead to gain of function in accelerator mutations, which are your oncogenes, and this leads to the formation of cancer. So the loss of function is very important here. Most mutations lead to loss of functions. Okay, very few mutations lead to gain of functions, essentially. But both in this story, both are bad for us. Now, this is the mutation hypothesis of cancer. How do we think about it in epigenetic terms? Like I told you, again, the central dogma comes to a rescue. And if we know that if we obstruct, the environment can obstruct this genetic information from being read, we know that we lose that functional protein. And if that functional protein is lost, essentially without creating a mutation, we are losing the function of tumor suppressor genes just by obstructing that genetic thing from get read. And how is that obstruction being done? By DNA methylating those promoters of tumor suppressor genes that result in this phenotype, right? So therefore, just by suppressing a gene or losing function by DNA methylation, by silencing a gene by DNA methylation, we are creating a mutated phenotype without altering the DNA, and that can eventually lead to cancer, right? Mutated genes and cancer. So what we know is that because different cancers are very different in their epigenetic phenotypes. Okay, like for example, a breast cancer will have a very different set of genes that are going to be methylated versus a colon cancer will have a different set of genes that are going to be methylated. And these come from, because I said that cancer is a developmental program gone awry, these methylations will happen as a function of that developmental program, right? So therefore, if I study these methylation marks very, very carefully, I can, just by looking at any random cancer, I can look at the methylation marks and predict which type of cancer this is. Not only that, within a cancer, we have seen that within the same cancer, there are multiple modules of DNA methylations that can exist. So for example, if I take just breast cancer, phenotypically, I can divide breast cancer into, say, 4 types: luminal A, luminal B, there's triple negative and regular, right? So there are these 4 types of breast cancers that you can histologically define. But then if I look at it epigenetically, it can be defined into many more types of cancers, because within those subsets, there are epigenetic differences. And epigenetic differences are very important to know, because every program will give a different output of gene expression. And depending on the output of gene expression of those cells, that reactivity of those cells to the drug will be different. So therefore, I can't take one drug and imagine that I will cure all types of cancer, right? So therefore, to understand the types of methylations that are there can give a very high prognostic as well as diagnostic basis for us to predict which drugs will be useful for what types of cancer, or which individual, even if you have the same cancer as the other individual, even in your cancer, what is special about your cancer in terms of your epigenetic phenotype compared to the other person's cancer. So these are the things that are now touted as personalized medicine. And these are currently being actively researched on.
Fantastic.
Subhojit, there's a question on Lamarck. If Lamarck's theory is right, then does that mean that somatic cell genetic mutation can also play a role in inheritance of disease?
No, that's not true at all, because somatic cells don't, at least so far, there's no evidence to suggest that. That is what the Wiseman barrier was. And that's what I explained before, that the Wiseman barrier will not allow the somatic mutations to translate, to sort of get into the germ cells, right? So what we know is that what affects the somatic mutations, right? What this environmental stresses, the same environmental stresses are affecting your germline cells as well, your stem cells as well. So these germline stem cells can get altered epigenetic phenotypes, and those altered epigenetic phenotypes can maybe affect mutations. So these are all maybes. We don't still have a strong theory for this. This is still a hypothesis in building. So we actively work upon these hypotheses in the lab.
I think Shantini ma'am wants to ask a question.
Sure.
Subhojit, I enjoyed your talk so much. It was so simple and lucid and explanatory. The feedback link would be shared to our audience, so please hold on for you to get your certificates. There's a question that we hear a lot of talk on epigenetics and how you can predict cancer, but I would like you to throw light on what other diseases is this field helping in detecting or maybe curing or whatever.
So right from, say, for example, even lactose intolerance. Why does lactose intolerance happen? Right? We know that we are all growing up drinking milk, but suddenly one fine day we decide, our body decides to reject milk, right? So even simple diseases like that, any genetic disease, if you look at the theory, the genetic basis of cancer that I showed, essentially it was undoing tumor suppressor genes. The mutations were undoing tumor suppressor genes, giving rise to loss of function of mutations. And what epigenetics, based on the central dogma, what epigenetics does is exactly the same thing. If you block gene transcription from happening, you will lose that gene function. So essentially, you can achieve the same mutated phenotype without mutating the gene. So every genetic disease that arises from loss of function can now be also looked at from an epigenetic angle. So it really, cancer is just one of those examples that we can really sort of carefully look at, because in a lifetime we can follow an individual and look at what happens. But essentially this goes for any disease that has genetic basis where there is a loss of function involved. Okay, there are also diseases where gain of function is involved, and that also there has been implications of epigenetics there as well. But the causal role versus the effect role of epigenetics is not that clear in those cases. Okay, and that is important for us to understand if you want to treat the disease. If it is causal in nature, then treating it later on after he has the disease is not going to help. So to understand whether epigenetics is causal in that disease versus effect in that disease is very important. And if it is causality that is proven in that disease, then if you think about it, we can actually treat the disease even before it happens. Once we develop markers for epigenetic alterations in, say, humans, for example, before, say, I showed that ROS can create epigenetic alterations. The reactive oxygen species can create epigenetic alterations that can change stem cells and make them more sort of push them down the cancer pathway, down that hill, right? But if I can develop markers now, biological markers that can test for these stem cells that are already not yet down the hill, but are on the hill and are about to go down that hill, down the cancer path. If I can develop epigenetic markers to analyze that, I can stop the disease even before it happens. And that is important. That is another hallmark of epigenetics that I didn't touch upon, is because all these modifications are reversible in nature. And therefore, because it's not a genetically written down, it's not fixed in our DNA, all these can be reversed. So even though we are at the fault of our grandmother's doings or whatever, some of this, if we understand carefully how to undo them, we can technically reverse some of these phenotypes as well, and disease predisposition as well.
Thank you, Subhojit, for such a wonderful explanation. I request our principal, Anita ma'am, who has a message for you, to come in. Hello, seems like the network there is having an issue, but actually she had messaged me that rarely does she sit through any microbiology lectures or sessions, but she was so interested and enjoyed your talk so much that she was spellbound. And to your credit, I must say that you're a speaker who had a backup of slides to answer so many questions. So I wish that all the audience would take a lot from you and acquire more knowledge in this field from you. Thank you so much, Subhojit. So Shweta, can you please take over?
Yes, thank you, Shantini ma'am. Thank you, Subhojit sir. We come to the end of this session. It was an enlightening and enjoyable experience for all of us here to get introduced to this new area of epigenetics. Due to time constraint, we will not be able to take in all the questions, but you can put up your questions on your registration WhatsApp groups and we will forward it to Subhojit sir. For the participants, the feedback link has been provided in the comment section of your YouTube. Kindly use the link which is also being put up in your registration WhatsApp group. So you can either do it from the comment section or your registration WhatsApp groups. This will help us to send your certificates more conveniently. Please ensure that you fill in the feedback immediately after we end the session, as the link will be open only for 15 minutes after close of the session. I now call upon Mr. Suman Gangar to propose the vote of thanks. Over to you, Suman.
Thank you, Shweta ma'am. Dr. Sain, questions are still pouring in. We are getting questions on private WhatsApp groups, but we have to end somewhere. And I am here to propose the vote of thanks. Participants, as Shweta ma'am has said, please post the questions in the group. We will definitely try to revert back with the answers. So on behalf of management and VS College of ASC, I would like to thank Dr. Subhojit Sain for accepting our invitation and delivering an excellent talk, or can I say an epi talk, in this webinar. Heartfelt appreciation to Mr. Shashank Joshi for the technical support provided for organizing this webinar. Gratitude to Principal Dr. Anita Kanwar and staff members of Microbiology Department for their efforts to make this event successful. Last but not the least, I would like to thank all the participants without whom this event would have not been possible. Stay safe, take care. Thank you.
Thank you, everybody.
Thank you,
Thank you,
Suman.
Subhojit.
I'm sure that Dr. Subhojit has put your thinking caps on, and you will read and think more about epigenetics. So I think we should now say it is all in our epigenomes, isn't it? Dear participants, we would also like to inform you that we are organizing our next webinar on Saturday, that is the 6th of June, for which we will soon send you the details. Thank you for being a part of this webinar. We look forward to interacting with you in the future. Thank you to everyone. Stay safe and keep your spirits high. Goodbye and be connected. Thank you.
Thank you to everybody.